Hello and welcome to the STEIL Dihybrid Cross Punnett Square Model Lesson Tutorial. In this video, I will explain the design, use, and additional considerations for the Punnett square allele models for Mendelian inheritance for determining the possible outcomes of crossing two genetic traits from parent organisms. The STEIL model was designed using a common shape for a chromosome as a way to tie the concept of genes and alleles to DNA, though it would be practical to use other shapes instead. The allele models were produced with three variable traits in mind to help students identify differences in alleles being represented. These varied traits were texture, size, and color. The color trait was chosen as an additional tool for students who can access the information using their vision, but it is not required for accessing the information presented in the model. One version of the model was made large with a rough texture and was printed using black PLA filament. One was produced large with a smooth texture printed in orange. One was produced small with a rough texture in violet, and one was produced small with a smooth texture in red. Small pieces of Velcro hooks were attached to the bottom side of each allele model to help it stick in place on the Punnett square backing, which will be described shortly. Additionally, on one end of the large allele models, a Braille D and R were included to give another method for identifying differences between the shapes. These can be used for Braille readers primarily working through a 2x2 single-trait Punnett square, or monohybrid cross, but the small allele shapes can be used for that as well. In this video, we are covering the use of the 4x4 dihybrid cross square, so the Braille labels are not necessarily relevant. A tactile Punnett square was produced to help students organize the model as they work through Mendelian inheritance heredity problems. This square was produced by measuring out and cutting wooden dowel rods to act as borders, then hot gluing them in place on a foam board to act as backing. Inside of each space in the square, and in positions on two of the outside edges aligning with the sections inside the square, Velcro loop material was attached to help keep the allele models in place. Braille labels for parent A and parent B were placed on the left side and top side of the Punnett square respectively. Before using the 4x4 Punnett square to do a dihybrid cross, each parent's genes need to be sorted into groupings to determine what alleles will possibly be donated to the potential offspring. This is because the dihybrid cross is meant to be used for determining heredity patterns for two traits at the same time. In this example, one of the parent's genetic traits show 4 alleles. 2 for one trait and 2 for a second trait. Let's say the two traits in question are for a pea plant height and flower color. We will say that the rough texture alleles will be for height, with the large piece being the dominant tall trait, capital T, and the small piece being short, lowercase t. The flower color will be represented by the smooth pieces, with the large piece representing dominant purple coloring and the small piece representing recessive white coloring, capital and lowercase p respectively. The parent's genetics being shown would then be heterozygous tall, heterozygous purple flower color, or capital T, lowercase t, capital P, lowercase p. The parent pea plant will donate one of the two alleles for each of the two traits to its offspring, but there are 4 possible options for which version of each trait will be passed on. I like to use the FOIL method for sorting the possible groupings of alleles when approaching a dihybrid cross problem. This is an adaptation of the same method used when multiplying binomials in math class. Sorting will follow the pattern first, outer, inner, and last. Combination 1 is dominant tall and dominant purple flower, capital T, capital P, or the first letters for both traits. Combination 2 is dominant tall and recessive white flower, capital T, lowercase p, the outer 2 letters. Combination 3 is recessive, short, and dominant purple flower, lowercase t, capital P, the inner 2 letters. Combination 4 is recessive, short, and recessive white flower, lowercase t, lowercase p, the last letter for both traits. Each combination would be set out in its own individual row or column on the Punnett square in the parent genetics area, and each box inside the Punnett square would get 2 alleles from both parents, resulting in 4 alleles to be inherited by the offspring. Continuing using previous example, we will add in a second parent donating capital T, lowercase p for all combinations, having homozygous dominant tall and homozygous recessive white flowers. The top left box will receive capital T capital P from our first parent and capital T lowercase p from our second parent, giving the combination capital T capital P lowercase p. The next box across the top row will receive capital T lowercase p from the first parent and the same from the second parent, giving capital T lowercase p to the offspring. The third box will receive lowercase t capital P from the first parent and again capital T lowercase p from the second parent, giving the offspring capital T lowercase p lowercase p. The fourth box in the top right will receive lowercase t lowercase p from the first parent and again capital T lowercase p from the second parent, giving the offspring capital T lowercase p lowercase p. In this example, I accidentally ran out of large rough pieces for the capital T and had to steal one from parent A to finish the first row. All three remaining rows are repetitions of these combinations, so we will not be going over the remainder of the 4x4 square. This is not necessarily the case for all problems that students may do using this model, but for brevity, I chose to use something that would repeat here. The final result from all 16 boxes would be an equal 25% probability of each unique combination being inherited by the offspring. A consideration to make when using these models, especially if printing a large amount of the model pieces is not feasible, is that because each piece has two tactile ways of identifying it, a single piece could be used to represent two different alleles for two traits at the same time. For example, the large rough piece could represent the combination capital T capital P, the large smooth piece could represent capital T lowercase p, the small rough piece could represent lowercase t capital P, and the small smooth piece could represent lowercase t lowercase p. This can reduce the number of pieces being used in the model, but this is recommended only for students who can keep in mind that a single piece represents two alleles instead of just one. One final consideration to make is that the size of the allele pieces can be scaled to adjust the amount of filament being used to produce them. This may be beneficial for reducing the amount of time needed to produce all of the pieces needed for the dihybrid cross. Thanks for watching the 4x4 Punnett square tutorial. We hoped this video helped with your understanding of how to use the Punnett squares for dihybrid crosses. As always, a link to the model files as well as a sample lesson plan can be found in the video description below.